Hello students, welcome to the first lecture of the chapter Biotechnology Principles and Processes. So let us first understand what do you mean by the term biotechnology. So traditionally it refers to the set of techniques that uses live organisms, okay, or their products like enzymes or other antibiotics, proteins, hormones, etc that these live organisms produce, okay, and we humans we find these products useful and we use it for various kinds of activities like making, for example, curd, or we can make bread or wine, okay. So all these products that are useful to us, they are actually made uh, with the help of the living organisms like microbes. And because these microbes produce certain kinds of enzymes or other kinds of proteins, okay. So these enzymes actually help us make these kind of products, curd, bread, or wine. Other examples could be making genetically modified organisms, okay. So this is bit advanced techniques that comes under the biotechnology, broad class of biotechnology. Then in vitro fertilization, through which people make test tube babies. So how the techniques are used, those all techniques come under biotechnology, as well as we can now create a synthetic gene, means the sequence of the DNA we can dictate, we can control and create a synthetic gene, as well as we can develop vaccines like DNA vaccines. So the techniques that are used for making this possible all comes under the broad class of biotechnology. So one can actually divide biotechnology into the traditional one, where or the conventional biotechnology, where people mainly focuses on the enzymes or other kinds of products that live organisms produce, and those produced enzymes, they're used for making something useful for humans like curd, bread, wine. And another class of biotechnology processes which can be termed as advanced, which helps us to do much more sophisticated things like making genetically modified organisms like plants, animals, that perform in vitro fertilization, synthesize gene and develop DNA vaccines. According to European Federation of Biotechnology, so this is a body in Europe, they gave a more formal definition for biotechnology. So what they state is the integration of natural science and organisms, cells and parts thereof, and molecular analogs for products and services. So what they mean is that everything natural, that everything available in nature, like organisms, plants, animals, even microbes, viruses, etc., and individual cells from these plants, animals, etc., and as well as part of cells like nucleus, mitochondria, or any particular protein, or any cytoskeletal element, anything, a part of particular cell, as well as molecular analogs, means DNA, RNA, of the molecules, carbohydrates, proteins, lipids, etc. So when you use these things, okay, integrate in such a way to produce products and services, obviously for well-being of humans, then that kind of way of utilization through techniques of biotechnology is what is said in this definition. Okay. Now in biotechnology, we can actually subdivide this very big field into two subfields. One is genetic engineering, another is bioprocess engineering. Okay. So these two actually are the main subfields. But today, biotechnology has evolved so much that there are hundreds other subfields that have all branched from these two principal subfields. Okay. So what do you mean by genetic engineering? Here, people utilizes techniques to alter genetic material. Okay. like DNA and RNA, and then introduce them into some host organism in order to change their phenotype. So they isolate some DNA or RNA from XYZ organism, okay, and then uh, introduce that artificially into another organism. Now, who organism who is receiving that DNA or RNA is called the host organism. Why do that? They do that to change the phenotype or the observable characteristics of the host organism. So there are set of techniques that makes this possible, and the study of these techniques comes under the broad field of genetic engineering. And now there is another subfield that is called bioprocess engineering. Now here, what people do, they grow desired microbe or eukaryotic cells like mammalian cells, plant cells, animal cells, etc. That too in very large quantities. Why? Because these cells individually have been discovered to produce some kind of a useful protein or useful product. Okay, and in order to produce that useful product, these cells or microbes, they are grown in very large quantities. So what kind of useful products that we are talking about? These include some antibiotics or vaccines or enzymes, etc. So if you know the penicillin antibiotics, so it is produced natively by a fungi, penicillium. So people actually then are growing this penicillium fungi in very large quantities in order to produce the antibiotic penicillin. So this is an example of bioprocess engineering. Now let us understand like why there is so much need to control uh, or to need to transfer genetic material into a host organism and then control its phenotype. Okay, why one needs to do that when in nature there is already a process called sexual reproduction exist. Okay, so you know what is sexual reproduction here. Get the two male and female gametes are formed, right? And they fuse to make a embryo that gives rise to a new organism, right? So when this process happens, there are process involved like recombination, etc., that leads to introduction of variations in the genome. So a few set of variations which are not harmful, but that is what makes the children bit different in characteristics from their parents. Even it leads to differences between siblings. So greater this so this variations, the introduction of this variation is made possible when sexual reproduction happen. But whereas when there is asexual reproduction, there is no such process like recombination, right? So because of which the genetic information is very well preserved. Progenies that are made as a result of sexual reproduction, they have a unique combination of genes, right? Due to the process of recombination. And now some of these combination of genes, they may be desirable and some may be undesirable. Okay. But when people are trying to create a hybrid, so genetically modified organisms, they only want those characteristics which are desirable. Like they want crops which are resistant to pest, resistant to drought, okay, or which gives fruits which have longer shelf life. So these kind of characters are desirable. But if due to just sexual reproduction, undesirable characteristics like much more susceptible to pest or susceptible to getting rot, okay, or the nutrient qualities is not that much. So if these kind of properties are coming about in the progenies, then people usually discard those kind of progenies. So there is a lot of trial and error involved in order to select out the desirable crops. So people want to make this process easier and pretty straightforward. So for doing that, people are using genetic engineering techniques, where first people has to create what is called a recombinant DNA, which I'll explain it later. And then the recombinant DNA is cloned. Okay, means cloning means multiple identical copies. Okay, they create identical copies of particular DNA piece. So that is called gene cloning. Once they have required number of clones, they then transfer into host organisms where they want this particular recombinant DNA to express. That is gene transfer. So these are three important genetic engineering techniques that helps us circumvent the problem of, um, having to see undesirable characteristics in the living organisms. We can only introduce the desirable characteristics that we want to see, and that gives us the useful products. So that is made possible with genetic engineering techniques. So what one does, they first identify a organism, say a organism, which have the gene or the DNA piece of DNA that codes for a desirable characteristics. This is the alien piece of DNA. This particular is the alien piece of DNA that they have isolated from host A. Then using some steps, they transfer it into another host, that is host B. Now will this piece of DNA, this alien piece of DNA, because for host B, this is something alien, right? This is something foreign. So will this foreign piece of DNA multiply in host B? Okay, because if it doesn't multiply, then we will not get clones, right? And if it doesn't express, if this alien piece of DNA doesn't express, then we won't see the desirable phenotype in host B. Now, uh, so we want these things to happen, but how will it happen then? So for this to happen, this alien piece of DNA, it needs to be a part of host chromosome. Okay, it needs to be part of host chromosome, because in the host chromosome, that is in the chromosome of host B, there is something called ORI, or origin of replication. So when if you recall the process of DNA replication, there you have heard the term origin of replication. So that is the start point from which the DNA replication starts. Okay, and that is the starting point of DNA replication, where the DNA polymerase comes and binds and starts polymerizing new DNA strands. So that is the start point. So if this alien piece of DNA gets somehow linked with this ORI, right? So this is ORI and this is your alien piece of DNA, referring it by A. Then what will happen? When DNA replication starts at ORI, it will go in the 5' 3' direction, and it will find this alien piece of DNA as well. So it will, the DNA mechanism, replication mechanism, RNA polymerase and other factors, they will replicate or copy the sequence of this alien piece of DNA, assuming that this piece of DNA is the host only. Okay, it is of the host only, and integrate this alien piece of DNA within the new genome of the host B. So all the daughter cells of host B, they will have this alien piece of DNA in their chromosomes. So when some promoters are there that is linked with this alien piece of DNA, it will allow transcription and then subsequently translation and then ultimately expression of this particular foreign piece of DNA. So I've already explained what is gene cloning. Gene cloning means creating multiple identical copies of any alien piece of DNA. Now, one of the prerequisites for genetic engineering, or one of the steps of genetic engineering technique, is to first create a recombinant DNA. Okay. Now I will discuss how would you construct a recombinant DNA. what all things do you need? So Cohen and Boyer, they were two scientists who constructed the first recombinant DNA in 1972. So they used something called vectors. Okay. So they used vectors. Now there are various kinds of vectors. Now one of these vectors is called plasmids. Plasmid DNAs are vector DNA. I'll actually, what are vectors? Vectors are something that acts as a delivery system, delivery guy to deliver the alien piece of DNA from, say in vitro, from a maybe petri dish or something like that into the host organism. So they act as a delivery guy. So these are the vectors. So one example of vector is plasmid. So you already have heard the term plasmid, but again, let me iterate what are plasmids. Plasmids are autonomously replicating circular extracellular extra chromosomal DNA. So they are autonomous. Okay, autonomous in the sense that they can replicate on their own without the need of chromosomes, host chromosomes, origin of replication area. Okay, they don't need that. They can replicate on their own. Also, they are circular DNAs. They are like this, whereas normal chromosomal DNAs, they are linear, and they are extra chromosomal, means they are not found, they are not linked to the chromosome DNA. They are a group of DNA that is a DNA that is different from the chromosomal DNA. Similarly, you must have heard that there is also called mitochondrial DNA, that is also having the similar property of autonomously replicating, and it is also a extra chromosomal DNA. But here in biotechnology, or in for constructing recombinant DNA, people mainly use plasmid DNAs. So what Cohen and Boyer did, they took plasmid DNA from the bacteria Salmonella typhimurium. Okay? So they took its plasmid DNA and digested it, means they cut the plasmid DNA using something called restriction enzymes. So when they did that, they they ligated, or they linked or joined a gene that is antibiotic resistant to this plasmid DNA. Okay, so this is the plasmid DNA. They have cut it using restriction enzyme. So because of which it has become like an open circle, like open U shaped. Okay. Now here the antibiotic resistant gene, like say if this is antibiotic resistance gene, it is ligated here using the enzyme DNA ligase. So a resultant vector will be something like this. Okay. Now this plasmid vector, which is carrying this antibiotic resistant gene, now it will be called vector because it is delivering this antibiotic resistance gene into another host, that is E. coli or Escherichia coli. So here this plasmid vector is transferred. Now since plasmid vector can replicate autonomously because it has its own origin of replication. It doesn't require Escherichia coli's chromosomal origin of replication, but it only needs the DNA polymerase enzyme from the host, that is from the E. coli. So in presence of DNA polymerase enzyme of the host, this particular plasmid vector can be cloned, means it can be multiplied into multiple pieces, like multiple uh, plasmid vectors or multiple copies of recombinant uh, DNA can be, made possible within the host Escherichia coli. So from this discussion, what did you understand about recombinant DNAs? What are recombinant DNAs then? Recombinant DNAs are nothing but vector DNA, okay, that is linked with your gene of interest. Let's write it as GOI. So here in this example, the gene of interest is the antibiotic resistant gene, which is ligated with the plasmid DNA. So this is your then recombinant DNA. Okay. Now this recombinant DNA is transferred into the host, Escherichia coli. Now using the DNA polymerase that is present in Escherichia coli, multiple copies of the antibiotic resistance gene is made inside the E. coli cells. So this is called gene cloning or genetic cloning. This is what Cohen Boyer did, and made the first recombinant DNA. So recombinant DNA can only be made using vector DNA. Now in this example, plasmids are the vector DNA. So they genetically modified the Escherichia coli. Okay, so genetically modified the Escherichia coli by adding a antibiotic resistance gene. So the first step in creating any genetically modified organism, you first have to choose what gene you want to introduce into your host. That is, identify desirable genes. Next, what you want to do, you want to just introduce this desirable gene into the host. So for introducing it, you have to first create recombinant DNA. Then only you can introduce this desirable gene into the host. And then you have to maintain this introduced gene in the host and make sure that this new gene gets transferred to its progeny. That is, you have to make sure that your gene of interest gets multiplied. That is, gene cloning is happening. And while gene cloning is happening, it is also getting transferred, that is gene transfer to the daughter cells of the cell that you have genetically modified originally. So there are so the technology, or you can say a group of techniques that is used to create recombinant DNA. They together are classified as recombinant DNA technology. So under this technology, people use certain tools. First tool that you have seen in the previous example is restriction enzyme, the use of restriction enzyme. So in this example, here restriction enzyme was used to cut the plasmid DNA. So you need a library of restriction enzymes, and you also need to understand how these enzymes are going to cut. Okay, what or what type of DNA sequences these enzymes are going to target. You need to have a complete knowledge of that. Then you need the polymerases or DNA polymerases for cloning your gene of interest inside the host organism. Then you also need the DNA ligases, which will help you ligate your foreign piece of DNA with the vector DNA. Then you will need vectors or the delivery guys who will actually help you deliver your gene of interest into your host organism. So in the previous example, it was plasmid, but there are other vectors as well. And lastly, you need a host organism where you can make sure that your recombinant DNA will be stable and will be cloned, and it will be transferred towards daughter cells. So understanding, now we will understand each of these one by one. Okay. First, let us understand what are restriction enzymes, like how they were discovered. Okay. Like how to choose restriction enzyme, etc. Now, restriction enzyme was discovered in 1963. People actually found two enzymes in E. coli or Escherichia coli. that were responsible for restricting the growth of bacteriophage. Okay, so you know bacteriophage are the viruses that that infect bacteria like E. coli. So people discovered that in E. coli there are two types of enzymes that exist which prevent this infection by bacteriophage viruses. So these two enzymes were then isolated and studied. So they found there was enzyme one type of enzyme, which could add methyl groups to the DNA. So here DNA refers to the bacteriophage DNA. Okay. So due to addition of methyl groups, that bacteriophage DNA will be will not be able to get expressed. Okay. So this type of you know uh, thing is called, or this type of process is called DNA methylation. Now another enzyme they found uh, is is that that is present in E. coli, that these enzymes could cut the bacteriophage DNA. They could just simply cut. So if the bacteriophage DNA was cut, again they will not be able to get expressed and cloned. So the infection will not be possible. So these enzymes which were able to cut the DNA, they are of prime importance to us. They are of prime importance to us for creating recombinant DNA. Now these enzymes were named as restriction endonucleases. So the first restriction endonuclease to be isolated was HIND2. Okay, the name is HIND2. These enzymes, they have a property that they can recognize a specific sequence of base pairs on the DNA. Okay, and this particular sequence of base pairs is called recognition sequence. And today there are over 900 restriction enzymes. So when this book was written, it was 900. Now there are even more. Okay, that people have isolated from different strains of bacteria, and they are all isolated from 230 over 230 strains of bacteria. So restriction enzymes are actually isolated from bacteria only, but and people have created even large, like more than created library for even more than 900 restriction enzymes. So as I said, a unique property of restriction enzyme is that they can identify a unique set of sequence base pairs. Okay, they can recognize a unique sequence of base pairs within the DNA, and this unique sequence is called recognition sequence, and it is different for different restriction enzymes. No two restriction enzymes share the same sequence. It is unique. Okay. Now each restriction enzyme can recognize unique recognition sequence, which I already told. How these restriction enzymes are actually named. Let's talk about that. They are named as follows. So this is one restriction enzyme that is called EcoR1. Here the first letter, it refers to the genus of the bacteria from which it is isolated. So it is isolated from Escherichia coli. So the Escherichia is the genus name. So from there E is taken. Then coli is the species name. So the second two letters corresponds to the species name of the bacteria from which this enzyme is isolated. That's why Eco R1 is taken from Escherichia coli. So E from Escherichia, CO from coli. Now these two, basically these three letters up to here refer to the name of the bacteria from which the restriction enzyme is taken. The next two letters refers to what? Now this R, it refers to the strain, that's name of the strain. Okay, so Eco R1 is taken from RY13 strain. Okay, so the first letter of the strain name is the fourth letter of this nomenclature of Russian enzyme, and this one, the Roman letter 1, refers to the order of isolation. Means in which what is the order of the isolation of this restriction enzyme from this particular strain of Escherichia coli. So there can be eco R2, eco R3, eco R4. So 1, 2, 3, 4 refer to in which order these enzymes were isolated by a scientist. So in this particular table, you can see there are other restriction enzymes mentioned apart from EcoR1 like BAMH1, HIND3, TAC1, okay, SMA1. So here you can see that like in BAMH1, B refers to the bacillus, this is the name of the genus. AM refers to the first two letters of the species name, that is Amyloliquefaciens, okay. H1. So H will refer to the strain name and 1 refers to the order of isolation. And for each restriction enzyme, there is a unique recognition sequence that is given here in this table. Now, nucleases. What are nucleases? Nucleases are basically a group of enzymes which can cut DNA. Okay, these are called nucleases. Now, their nucleases are of two types. Like we can distribute nuclease into two subcategories, and it depends on where the cut is made. Where does this nuclease enzyme makes a cut within the DNA sequence? So there are exonucleases and endonucleases. Exonucleases, they have the tendency to cut at the ends of the DNA strand. So if this is your double-stranded DNA sequence, the exonucleases will have a tendency to cut towards the ends, like towards the 5' or 3' ends. It will not cut uh, towards the center of the DNA sequence. Whereas the endonucleases, they have the tendency to cut within the DNA strand, and that too at a specific site. So endonucleases, they can actually scan through the sequence, DNA sequence, and wherever it finds a specific, it's unique and its specific binding site, there it will bind and it will cut the DNA strand towards the middle, not exactly middle, but towards the middle of the DNA sequence. So these are two types of nucleases. Exonuclease, exo means towards the end, endo means towards the center. Now, how does restriction endonucleases function? How do they work? First, what they do, first and foremost thing, is that they scan through the entire length of the DNA sequence, okay, to look for its specific recognition sequence. First, it has to find where is its recognition sequence present within the entire length of DNA sequence. Once it finds its recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific points in their sugar phosphate backbones. Okay, so after it has found its recognition sequence, it will go ahead and bind to the DNA, and simultaneously it will cut both the DNA strands, the double helix structure. Okay, and that too at specific points. What are those points? I'll come to it later. And it cuts in such a way that the backbone of the DNA is cut. Okay, sugar phosphate backbone is cut. So in this way, it can create DNA fragments. So restriction enzymes again have a unique property, is that they can recognize palindromic sequences. Okay, they can recognize palindromic sequences within the DNA. So what are palindromes? Palindromes are basically words which reads the same when you read backwards as well as when you read in the forward direction. Okay, for example, uh, madam. Okay, So when you read from this direction, it reads madam. And when you read from this direction, here also MADAM. Here also it reads the same. This is the palindrome. So in English, this is a palindrome, example of palindrome. Whereas in DNA sequence, what does palindrome mean? Here also the logic applies. So for the restriction enzyme EcoR1, this is its recognition sequence. Okay, here is its recognition sequence. So when you read this sequence from 5' to 3' direction, it is GAATTC. Whereas when you read from 5' to 3' direction, here also GAATTC. So it reads the same when you keep the orientation of reading same. Whereas when you read from 3' to 5' direction, like CTTAAG, here also in this strand, when you read from 3' to 5' direction, CTTAAG. Here also the sequence reads the same. So you have to keep the orientation of reading same in order to get the same sequence. Okay, so that is the palindromic sequence. So recognition enzymes, these restriction enzymes, they can only recognize palindromic sequences. Means their restriction sites or recognition sequences, they are palindromic in nature, and they have a tendency to cut little away from the center of the palindromic recognition sequence. They do not cut exactly at the center. Like this is a 6 base pair sequence, so it will not cut here. Okay, it will not cut at the center. It will have a tendency to cut towards either this end or towards this end, means towards the ends. Okay, but wherever it cuts, it is same. That point is same for both the DNA strands. So if it cuts between G and A, if EcoR1 cuts between G and A in the strand that is running from 5' to 3' direction. So in the second strand, but it's running from 3' to 5' direction, here also the EcoR1 will cut here between G and A. This point of cut is same for both the DNA strands, but it is little away from the center. That's it. And when it cuts, it creates something called sticky ends, which are nothing but single stranded ends. So here, this is the recognition sequence for the restriction enzyme EcoR1, like GAATTC for 5' to 3', and here also from 5' to 3'. Now this EcoR1 cuts here and here, okay, between G and A and between G and A. So when it cuts, what happens? Actually, the cut will be here between G and A. So when it cuts between G and A on both the strands, this particular section gets removed. Okay. And here also this particular section gets removed. What is the remaining section then? It is this AATTC. Okay. And here G will be there. So this is your sticky end, because the sections that were here and that were here, they are removed by the EcoR1 enzyme, leaving behind this kind of a sticky end. So as you can see, the base pairs A, A, T, T, they are alone. So they can now easily form hydrogen bonds with bases which are complementary to it. So it can form hydrogen bonds with another sticky end, which may have the sequence like TTAA, right? And it can, here it can come C and G, something like this. If it comes, then it will complete the region that was previously lost by the action of EcoR1. So for creating sticky ends which are complementary to each other, one sticky end will be present in one DNA, another sticky end will be present in another DNA molecule. So for creating this kind of complementary sticky ends, one must cut both the DNA molecules with the same restriction enzymes. So this kind of um, complementary sticky end from another DNA molecule can only be created if both the strands are cut by the same enzyme EcoR1. This will become more clear from this particular figure. So here, this is one DNA molecule. It is also having the recognition sequence for EcoR1, right? And this is another DNA molecule which is also having sequence for EcoR1. Here it cuts like this, okay? Cuts like this between G and A. So after it has cut, these portions, they are removed. Here also these portions are removed, leaving behind two sticky and one, and this is two. Now these two sequences, the overhanging single-stranded DNA sequences, here AA, TT, and here TTA, they are complementary to each other. So they can now form hydrogen bonds with each other, okay, and create this kind of a molecule. Now, if suppose first DNA molecule is your vector DNA, like a plasmid DNA, and second DNA molecule is your gene of interest or the desirable gene, which you want to introduce because it will give some desirable characteristics in your host organism. And both are cut with the same restriction enzyme, EcoR1, leaving behind similar kind of sticky ends. So uh, these sticky ends will now bind to each other by forming complementary hydrogen bonds. And these hydrogen bonds can be sealed by the action of the enzyme DNA ligase. Okay, DNA ligase has now sealed these sticky ends with each other. So here DNA ligase will do its action in the backbone. Okay, here hydrogen bonds are formed, and here DNA ligase, referring by D, they have now uh, formed the phosphodiester bond at the sugar phosphate backbones. So it will now create one long piece of DNA where some portion is from plasmid and some portion is from the foreign piece of DNA. Now this complete DNA is now called the recombinant DNA. So recombinant DNA is nothing but a foreign DNA that is linked with the vector DNA. And for creating this kind of DNA, you need restriction enzyme and DNA ligase. Now another topic that is very useful is knowing the technique that is called gel electrophoresis. That is also called agarose gel electrophoresis. Okay. So what is this technique? So when you have cut your DNA by restriction enzyme, it will result in DNA fragments. So suppose you have a genome, say a very long genome like human DNA, human genome you have, and you have cut it using restriction enzyme, one restriction enzyme, EcoR1. So since EcoR1 will, scan through the entire length of DNA, wherever it finds is 6 base pair or recognition sequence, it will cut. So you can actually assume that the genome can be cut into multiple fragments. How many? You don't know yet. Okay, you don't know. Since you cannot see the process happening clearly in front of your eyes, the process is not visible through naked eyes. It will be like all the reaction is happening in a very small volume of solution. So you need to visualize those cut DNA fragments. So how you will do that? For doing that, you need to first separate it out, okay, from each other. And how the separation should happen? They should happen based on their lengths. Means the smallest DNA fragment should say come at one end, the largest should come at the other end. So for doing this kind of sorting of the DNA fragments based on their length, you need a technique that is called gel electrophoresis. Okay, so how this technique works, you know that DNA, backbone of DNA consists of phosphate groups. So because of which the complete DNA molecule has a net charge, has a net negative charge. Okay. So when you apply an electric field where there is one end is anode, another end is cathode. So can you guess, uh, if you place your DNA molecule as at the cathode end, where it will move? If you are assuming that it will move towards anode, you're correct. It will move towards the positively charged end because it itself is negatively charged. So under an electric field, when DNA fragments are present at the negatively charged end or the cathode end, DNA molecules, the DNA fragments, they will slowly migrate towards the oppositely charged end. And when it does, it will, separate out. How it will separate out? People actually use a porous matrix, and this porous matrix is actually a agarose gel. It is a polymer basically that is extracted from seaweeds. So this creates a gel matrix, a thick block of gel. So, and this gel will have pores of various sizes. So the DNA fragments which are smaller in size, they will naturally have the tendency to move faster through the pores. Okay, but the DNA fragments which are longer in size, they cannot migrate as much faster through the pores as the smaller ones. So what happens uh, ultimately? So if this is the negatively charged end, this is your positively charged end. Okay, and this is your agarose gel matrix, the gray colored. So at the wells, people create small wells. Okay, within these wells, they load their DNA solution, which is having DNA fragments of various lengths. So under the applied electric field, here electric field you have applied using electrodes. Here what happens? The DNA fragments which are quite large in length, they do not have the ability to move very fast through the pores. It will the pores will restrict its movement, because of which the large DNA fragments, they will form separate bands towards the cathode end. These are the bands which are larger in size. Whereas the small DNA fragments, for them the path is much more smoother. So they will easily move towards the anode end and will create bands towards the anode end. So in this way, the DNA fragments, they are distributed from the largest to the smallest. Okay, enough. So it creates, so when you visualize this, it creates an image like this. Okay, where the largest bands are here and the smallest bands are here. So people can actually mark, like this is your 1000 base pair band, this is a 700 base pair band, and you got a band which is in between 1000 and 700, so it is 800 base pair. So in this way, bands are made and people have visualized it. So how they visualize it? They actually visualize it using a carcinogenic compound called ethidium bromide. So while handling this particular chemical, people have to be really careful, uh, and like they have to wear gloves and make sure that this compound doesn't touch their skin, as well as this compound has to be disposed of very carefully. So they stain the DNA fragments with this, after which the stained DNA fragments which have formed bands, it is exposed to UV, because this ETPR stain, it uses UV radiation to emit light, emit color. Okay, so these bands, this band that you're saying in gray and white. So this is possible after staining with ATBR and then giving UV light. So there are particular machines that do it in single step. So after people have visualized it, so here in this case, people have got bands which are of, or people have got DNA fragments which are of 800 base pairs. Now, if that is their desired piece of fragment that they were looking for, that at the beginning of the experiment, they knew that somewhere here they would find the DNA fragment of their interest. What they will do, they will cut it out. They will cut out the band from the agarose gel and then dissolve out the agarose gel, leaving behind only the DNA fragment. And then DNA fragment will be, separated out through small filters, okay, from the solution, agarose gel solution. Okay, this process is called elution. So through elution technique, this particular piece of, say, DNA fragment can be eluted out or can be separated out from the entire block of agarose gel. Now, coming to the next topic, till now, you saw water restriction enzymes and also you understood the technique of gel electrophoresis. Next, let us understand the vectors, which are the delivery vehicle for the transfer of your desired piece of DNA into your host organisms. So there are two types of vectors basically. One are cloning vectors and another are expression vectors. Okay. Cloning vectors, as the name suggests, it will help in making multiple clones of your gene of interest. Whereas expression vectors, they will help in expressing your particular gene of interest. So this you will study in your higher classes. But in this chapter, we'll mainly focus on cloning vectors. So mainly two types of cloning vectors are being used in a biotechnology, in the field of biotechnology and genetic engineering. So one are plasmids, another are bacteriophages. They are mainly used. Why? Because they have the ability to replicate within bacterial cell independent of the control of chromosomal DNA. Means the plasmid DNA or the bacteriophage DNA, they do not need to be integrated with the chromosomal DNA, okay, in order to get replicated, in order to create multiple copies within the bacterial cell. What they need? They only need the DNA polymerase enzyme of the bacterial cell in order to replicate. Otherwise, the origin of replication sequence is present in both plasmid DNA as well as the bacteriophage DNA. Now, bacteriophages, uh, now the choice of their vector, whether you will choose a plasmid or whether you will choose a bacteriophage, also depends on how many number of copies or how many number of clones you need for a particular gene of interest or a foreign DNA. If you want a large number of clones, okay, of your gene of interest, then maybe you should go for a bacteriophage virus. Okay, means link your foreign piece of DNA into the bacteriophage's DNA, their genome, and allow bacteriophages to infect your bacterial cell. Or if you are happy with only few number of copies of your gene of interest, then maybe go for plasmids, which now among plasmids, there are also a variety of plasmids that is available. And there are few plasmids which can create only one or two copies of a gene of interest. And there are also few plasmids who can create 15 to 100 copies, means a large number of copies. So it depends. So your choice of vector of your, or the choice of your cloning vector, basically, it will be one of the factors that will help you, choose a cloning vector is how many number of copies of foreign DNA do you want. So gene cloning, what happens? So if you are able to link your foreign piece of DNA with bacteriophage DNA or plasmid DNA, then it will multiply. Obviously, its number will multiply equal to the number of copies of the plasmid or bacteriophage. Obviously, when the DNA replication will happen, it will not just happen for a particular gene of interest, but it will happen for your entire recombinant DNA. Okay, that is plasmid DNA plus foreign piece of DNA, bacteriophage DNA plus foreign piece of DNA for the entire uh, recombinant DNA. You will get multiple copies, and in each copy you will have your foreign DNA or piece of your gene of interest. Now, cloning vectors, uh, what are the properties of cloning vectors? First, they should allow easy linkage of your foreign piece of DNA. Okay, you do not have to worry too much because about linking. It should be pretty straightforward. Next, it should allow easy selection of recombinants from non-recombinants. Means since this process is not that much visible through naked eyes, so you need particular assays in order to make sure that your host cell in which you have introduced your recombinant DNA, okay, is it that particular host cell or not? Because all the cells superficially will look exactly the same. You cannot distinguish it that which of the host cell will have your foreign piece of DNA. So you need some easy way of determining or easy way of sorting out the cells with your recombinant DNA and cells without your recombinant DNA. So these two things should be quite easy. Means since they are the basic steps, they have to be easy. The complicated jobs are, as I've already told you, first you have to select your desired piece of DNA. Now that is a complicated job, okay? And then introducing. But the techniques that are now developed for introduction of recombinant DNA into host cells, they're also pretty easy. But the first job, foremost job, is we have to find out which piece of DNA do you want to integrate. That is a complicated job. The next steps or genetic engineering, they're relatively easy. Now in this figure is given a plasmid DNA named PBR322. Now this plasmid DNA is originally taken from the E. coli bacteria. So in this figure, you can see few regions. Okay, first is the ORI or origin of replication, from where the replication process happens or the starts or it starts. Next, there are antibiotic resistant genes called amphicycline resistant gene AMPR and tetracycline resistant gene or TETR. Okay, these are resistant genes. And within the antibiotic resistance genes, there are recognition sites. Here it is also called cloning sites or restriction sites. Okay, there are multiple names referring to the same thing. Okay, so there are these sites that are present within the antibiotic resistant genes. Also, it is present in other region as well. So this region is separate from the antibiotic resistance gene. And lastly, there is another region called ROP gene. Okay. Now this ROP gene is actually needed for replication for this replication of this particular plasmid DNA. And within this ROP gene sequence, there's another restriction site that is PVU2. So this is how a typical plasmid looks like. So we'll discuss in the next class about each of these, structural elements of the plasmid DNA in more detail, as well as cover few more topics of this chapter. Thank you.